So our first presenter today is Brooke Gauthier. She's presenting her proposal on oak regeneration. Brooke came to University of Kentucky from University of New Hampshire, where she got her Bachelor's of Science in Environmental Conservation Sustainability with a minor in Forestry, Wildlife, and Conservation Biology. She has some research experience in this field from her time at UNH, including working on a regeneration survey of red oak and a Shelterwood harvest cut. So with that, I will hand it over to Brooke for her presentation.
Hi. So like Steven said, I'm Brooke, and I'm trying to share the slide. Is it sharing? I think it is.
Yes, we can see your computer.
Okay, sweet.
Okay.
And
Perfect.
put that over here. Okay, so my thesis is an 8-year regeneration response to gap-based oak shelterwood practices in Berea College Forest near Berea, Kentucky. Okay, so here's a little outline of what I'm going to discuss with you guys today, including an introduction and the experimental plan. So let's delve into the meat of it. So for a little bit of history about oak dominance, for most of the 16th and 17th century, oaks dominated the canopies across eastern North America, and during this time, Native Americans occupied the land using periodic fire that favored shade-tolerant and fire-tolerant species like oak. And then in the 18th century, European fire frequency became way too high for oaks to regenerate. This trend really continued, and fire was used intensively to clear the landscape and promoting species renewal in the 19th and prohibiting species renewal in the 19th and 20th century. And then because of this and the increase of wildfires due to the increase of European fire regimes, fire suppression began in the 20th century, starting with the Weeks Act of 1911, which appointed the Appalachians as a federal forest and promoted the lack of fire use. So then in the 1950s, people started to notice that oak regeneration was lacking compared to the 1930s. And this concern was due to the importance of sustaining the oak supply for timber harvesting. So because of this, research really began to take off, focusing on reduction of seedling competition, integrated control of competing regeneration in the overstory and understory effects of deer browsing on oak regeneration. So this kind of leads us to a present day where oaks are being underrepresented in they're still pretty dominant in the forest canopy, but they're being underrepresented in forest regeneration. So, and just to talk a little bit about composition shift, in Southern Appalachia, it is estimated that red oak regeneration is at about a 10% survival rate into the canopy due to the increased density of shade-tolerant, fire-intolerant species that are outcompeting the oaks. So this includes red maple, yellow poplar, and from this came the mesification hypothesis, which predicts that shade-tolerant, fire-intolerant species are replacing the shade-intolerant, fire-tolerant species. So red maple is outcompeting oak. And prior to the mesification hypothesis came Fee and Steiner's paper, which conducted a study researching whether or not maple growing stock is increasing in abundance and size class structure throughout its natural distribution in the 1980s through 2006. So they looked at 37 states, which you can see in this figure on the slide. And 26 out of the 37 states saw an increase in red maple in abundance and size class from 1980 to 2006. And red maple is also expanded into the Midwest United States, which it did not formally occupy. So that's a little bit of an issue. And then getting into some silviculture in order to promote oak regeneration, especially against such like hardy red oak competition. Many silviculture practices have been employed. So in 1986, a clear cut was done and promoted as unsustainable for promoting oak regeneration. In this 46-acre mixed hardwood stands that consisted of 53% oak prior to tree removal. So in table 1, you can see that the total number of stems presented hasn't changed much over 2 years, but species composition has drastically changed. So total stems only changed about 1,000 seedlings, but oak is only about 28% of the pre-harvest number. And then another unsuccessful regeneration story is single tree harvest. And this cut produces overstory light similar to an unmanaged mature hardwood stand, which is insufficient for shade intolerant species growth. So Shelterwood has been giving a lot of mixed signals, which have been deemed unsuitable for oak regeneration, but I do have a successful Shelterwood story to explain. So Laka et al. Examined light availability and species regeneration outcomes on 51-year-old gaps in 3 different gap sizes. So that is a 0.02 hectare, 0.16 hectare, and a 0.46 hectare gap. And by looking at these 2 different tables, you can see that the 0.2 hectare gap size had the smallest overall density with no saw timber volume and the smallest diameter at breast height for maple and oak, and the lack of density is most likely due to the effects of the surrounding forest, which is blocking sunlight for entering the small gaps. So these small gaps aren't really very helpful for regenerating much of anything, especially not oak. And then in the 16 hectare gap, this had the highest density of trees per acre and the largest relative density of oak. And then lastly, the 0.46 hectare gap had the largest saw timber volume and the largest basal area per meter squared per hectare, along with the highest density of yellow poplar. So just looking between these 2 different graphs, you can see that maple had a higher relative density than oak in all 3 of the gaps, showing that red maple is a strong competitor against oak, but the silviculture is successful in regenerating oak. So it's something to think about. And then the last silviculture paper I wanted to discuss with you guys is Hackworth et al., which recommended Shelterwood silviculture as it augments understory light availability for oak regeneration while inhibiting the regeneration of competitors. So they used a mid-story removal, which increased light availability by 10 to 20% full sunlight within the cut. And later it was found that the mid-story cut was able to produce white oak produce more white oak reproduction than the control plots, and that the removal had positive effect on white oak and red oak seedling heights on intermediate sites. So good news for oak. And then we're going to get into the objectives of my project, which are, the first objective is determining spatial gradient of oak and non-oak regeneration on location from the center of the gap. Objective 2 is determining spatial location of oak and non-oak regeneration based on a matrix with or without mid-story removal. And then objective 3 is comparing and contrasting the data collected in 2020 and 2014. And the need and significance for this, there is a gap between the understanding of long-term relationship between gap silviculture and spatial distribution of oak regeneration. So this is one of the longer oak studies that has been conducted. And oak is really important for wildlife, whether that is caching animals or deer browsing. And as we all know, they need white oak for bourbon barrels to promote Kentucky's economy through the bourbon industry. So if you're not a wildlife person, maybe you like bourbon, and this is important to you. So long before I was part of this project, Clint Patterson, who is now the college forester at Berea College began his master's under Dr. Laka, and created this Oak Regeneration project together and cut 12 gaps into Berea Forest in 2012. And these sites are established within a 3-meter radius between each other on the western edge of the northern Cumberland Plateau. So these 3 sites are Water Plant, which has 2 gaps, Pig House, which has 2 gaps, and Horse Cove, which has 8 gaps. And the dominant species within this canopy currently is a mixture of oak, hickory, ash, sugar maple, and yellow poplar. Here's a diagram of the gap cuts. So the inner white circle is the gap, while the blue outer ring is the area considered to be the matrix. And the inner gap has a radius of 30 meters, while the outer ring has a perimeter of 30 meters. So this is a total of 60 meters across the gap, or 60-meter radius. When the stand was cut, all trees greater than 1.27 centimeters in diameter at breast height and or were over 137.16 centimeters in height within the gap were removed. And then 6 gaps out of the 12 got a mid-story removal cut, while 6 gaps got no treatment, leaving the mid-story as a control. And then transects were established every 60 degrees using azimuth compass reading and placing pink wire flag at every 10 feet or 3.048 meters. So a steel fence post was established into the center of each plot, and then another rebar was established at 30 meters, and then rebar was established at 60 meters on each transect. And here's a photo of what the transects look like. It's a little hard to see, but you can see that there's pink flagging, and that's the transects that we're following. And then in 2013, 1-year-old rootstock white oak seedlings obtained from the Kentucky State Nursery were planted at 1-meter intervals in every gap, and a starting transect was randomly selected, and then every other transect following received a planted white oak seedling. So only 3 out of the 6 transects within the plots got white oak seedlings, and these seedlings were tagged with blue whisker tags and a metal label where the seedlings were planted. And then in September 2020, Zach Hackworth and I started to began reestablishing transect lines and began the Oak Regeneration surveys in October 2020. So beginning at the center of the gap, we walked each transect, scanning for oaks existing within 1 meter on each side of the transect. So essentially the transects are 2 meters by 60 meters. And during this inventory, we measured total height, species, and distance from the center of the gap. And distance was measured using a transponder. So this is what it looks like. We have a little remote, and we push the button, and it uses radio wavelength to connect with the yellow part on that pole, and it tells us how far away we are from the pole. So that's how we've been gauging how far away from the center of the gap we are. And then here's just a little diagram of all the transects. And then we began non-oak regeneration surveys in December of 2020. So the plot centers for 2 circular non-oak regeneration surveys were established every 10 meters along the transects for each gap. And so this totals 444 non-oak regeneration plots. Regeneration less than 1.5 meters will be conducted in a millacre plot, so that's 1/1000th of an acre, or a 1.13 meter radius. And regeneration taller than 1.5 meters will be conducted in a 1 out of 445th acre, or a 1.7 meter radius plot. And within these plots, height, species, and distance from the center. Or no, we're just doing height and species are recorded. So to get on to objective 1, the hypothesis that we are looking for is oak regeneration density and regeneration height will be the lowest at the center of the plot and increase as distance away from the gap increases, while non-oak regeneration density and regeneration height will be highest in the center of the gap and decrease as the center of the gap increases. So we think that oak will be less abundant in the non-oak regeneration will be highest in the center of the plot and decrease. So we think that oak regeneration will be less abundant in the center of the gap due to competition with fast-growing non-oak species such as the red oak and yellow poplar. Oh, yeah, and then for data analysis, we will be discretizing transects into 8 distance categories, which will be assigned in coordination to location treatment factor. So location treatment will be corresponding with the distance category located along the matrix treatment of the experiment, and these levels will be called gap and the mid-story removal and control. So distance categories located within the gap will be assigned a location treatment of gap, and distance categories in the matrix will be assigned matrix or control depending on whether the matrix got that mid-story removal or was left as a control. And then distance categories will be assigned regarding to gap edge. So D0 will be comprised of distances less than 3 meters from the gap edge. D1 is 4 to 12 meters, D2 is 13 to 21, and D3 is 22 to 30 meters. And this is again retrospectively from gap edge. So sample values of metrics of interest will be calculated for each experimental unit by average metrics measured in the 1/1000th acre and the 1/445th acre non-oak regeneration plots, contained with retrospect distance category location treatment combined along all of these transects. So there's a lot of data analysis going into this project, so it could be a little confusing. So for objective 2, the hypothesis is that oak and non-oak regeneration height will be the highest in the mid-story removal matrix compared to the control matrix due to better light availability. So to detect the presence of spatial patterns in pre-treatment data that may confound interpretation of post-treatment results, we will evaluate the differences in natural oak reproduction height and density in 2020 using a linear mixed model framework. So to quickly explain this, because again, there's a lot of data analysis involved in all of these objectives, oak height and density will be evaluated separately as response variables in linear mixed effects models with distance categories location treatment and distance times location treatment two-way interaction as fixed effects. So a choice of random component will be determined via Alkaline's information criterion, and the likelihood ratio test between the two models and the selected model will be used as a base non-spatial model. And then to evaluate the model adjustment due to spatial autocorrelation, we'll perform the model independently of 5 spatial correlation structures, which will be exponential, Gaussian, linear, ratio, and spherical. And then per the methodology of Zur et al. In 2009, we will calculate the Acknowledgements information criterion for the 5 candidate spatial models. And finally, the outcome of analysis will be deemed the final model, and the final model will be evaluated using a type 3 analysis of variance or type 3 ANOVA to elucidate significance of model factors, and Tukey adjusted pairwise comparisons of estimated marginal means will be employed to analyze differences among levels of significant factors. And then finally, we are on objective 3, which is oak seedling abundance we are hypothesizing that oak seedling abundance has decreased from 2014 to 2020 within the gap due to competition with early successional shade intolerant non-oak species, again, such as yellow poplar, red maple, and mean height and tree density for all oaks and white and red oaks separately as well. As for the 3 tallest trees per subplot will be analyzed using a repeated measures ANOVA to test for any differences in 2014 data collected and 2020 data collected. So this includes seedling height or density. And the ANOVA will also utilize the nested mixed model 1, which I described for objective 2. And then 2 years post-treatment, so this is 2014, and 8 years post-treatment, 2020, tree density for all seedlings and all yellow poplar and red maple seedlings separately will be analyzed using analysis of variance, so again in ANOVA, to test for significant effects of positive category and mid-story removal, position category. And then here was our tentative timetable, which isn't exactly accurate at the moment. We are hoping to finish in December 2020 for the non-Oak Regeneration survey, but we are still conducting that. And we have finished all plot setup and Oak Regeneration surveys. So we are a little more than halfway done with the data collection right now. And then just as some thank yous, I would like to thank Dr. John Locke for accepting me as his advisee and providing comment and help towards data collection. Zach Hackworth for helping with data collection. He's definitely been such an amazing help, and I definitely would not have gotten as far without him. Stephen Nevels for also helping collect data and for being my principal reviewer in the 601 class where we created our proposals for our thesis projects, and then my committee members, Dr. Stringer and Dr. Moeller, along with the rest of the 601 students, and Dr. Price for teaching that class. And then here are the literature cited.
Great. So Stephen Nevels is up next. He graduated from Ohio State University with a major in agribusiness and applied economics. During his time at OSU, he worked as a research assistant on a pollinator conservation project, during which he gained experience performing cost-benefit analyses. So that experience combining economics with conservation set him up perfectly for his current master's thesis project on maple syrup production in Kentucky. So Stephen has put in a lot of long hours in the field and in the office to get this project off the ground, and I'm excited to see how it's coming along. So without further ado, I'm going to turn it over to him.
Thanks for that, Ben. So I will start sharing my screen here. So yes, my project is a biological and economic analysis of maple syrup production in Kentucky. Start with an overview. I'm going to delve into the literature review, outline my research objectives and their respective methods, go over the preliminary work, then talk about the prospective timeline, and then the significance of the research. So starting with the production potential of maple in Kentucky. Kentucky is outside of the traditional range of maple syrup production, which is usually thought of as the Northeast. But the industry still has great potential here in Kentucky. There's estimated to be over 18 million viable taps in the state, which is more than enough for to support an industry. Additionally, the sugar maple tree is not the only maple that can be tapped for maple syrup. The red maple is also a viable option, which is important for Kentucky as we have a larger density of red maple in our stands compared to sugar maple. The maple syrup industry of neighboring states has been shown to be profitable. States like Ohio, West Virginia, and Indiana have growing industries, and I think Kentucky can also get in on this. Ohio has an estimated 400,000 taps, which generates over $4 million in production value. Looking at the biological processes that affect sap and sap production and sap sugar content, the sap yield and sugar content is impacted by a wide variety of variables. including climate and soil. The most obvious that will impact sap yield is the weather, as freeze-thaw cycles are necessary for sap runs, as this creates pressure in the xylem, moving the sap up and down the tree, which it'll then encounter the taps and then flow out. Soil nutrients also have an effect on sap sugar content. While sap yield is primarily dependent on the freeze-thaw cycle, the sap sugar content is dependent on the availability of non-structural carbohydrates. So prior a season of high masting in the fall will likely result in a lower sap sugar content in the spring, as a lot of the carbohydrates were used by the tree in that masting event. Crown characteristics and the sap sugar content have a positive value, which is intuitive because a larger crown has a greater photosynthetic potential, which means more carbohydrates and sugars can be produced for that tree. Moving to evaporation, which is how the sap is processed into syrup. Coming out of the tree, sap is around 2% sugar, and that's a decent percentage. Some can be as low as 1% or even below that, and some are higher, but a good average would be about 2%. But syrup has to be concentrated to around 68%, and so a lot of excess water has to be boiled off, which requires a lot of fuel to do this boiling. Fuel is estimated to cost at or over 35% of total production costs. And so small inefficiencies in evaporation can aggregate into larger costs for the producers. A way to get around this is reverse osmosis, which pre-concentrates the sap prior to going into the pan for evaporation. This significantly reduces the amount of water that needs to be boiled off. But there are limitations to reverse osmosis. The percentage that the sap can be concentrated to is estimated to be about 21.5%, but in my experience, that's much higher than the average producer would use their reverse osmosis for. Moving on to economic impacts, the direct impacts of maple syrup on the economy will depend on the total number of taps and the gallons of syrup produced. But one reasonable estimate highlighted in this chart is over $3 million, and this assumes 503,000 taps. The highest estimate is over 18 million, but this assumes a tapping rate of 2.94%, which is equal to that of Vermont, which is the highest producing maple syrup state in the nation. An estimate using implant software for an operation in Illinois offered about $9,500 in commodity value for 1,000 taps on a 20-acre stand with a 1.94 multiplier effect. And this illustrates that the impacts from increased maple syrup production will go beyond the industry and affect the economy at large. These two maps show the per county income alongside with forested acreage. The top map shows the income, and as you can see, the less affluent counties tend to be in the eastern portion of the state, and that is also where the higher density of forest area is. So this helps illustrate an opportunity for economic stimulus from maple syrup production and agritourism activities related to that. Moving into my project questions, I have 4 of them outlined here, and we'll go in depth to each one. Starting with the first, what is the relationship between physical and environmental characteristics in maple sap production and sap sugar content? We have 75 red maples and 75 sugar maples across 4 sites for a total of 150 trees. The trees were tagged with metal identifying tags and had their GPS locations recorded to ensure continuity across the years, with 2 hobo micro weather stations installed at each site, one on the north facing slope and one on the south facing slope, to record temperature data throughout the season. The trees chosen are representative of a range of DBHs, heights, and canopy levels. The mean DBH for the red maples is 33.5 centimeters, and the mean DBH for the sugar maples is 35.6 centimeters. Each tree will have the total height, crown depth, and crown width recorded using a laser rangefinder. And moving to the tapping, the trees have been tapped with a 5/16 inch plastic spile and polyethylene tubing used as the drop line leading down to a 5-gallon collection bucket. The measurements occurred from January to March of 2020 and 2021, which is the usual tapping season in our climate. Sap yield and sap sugar content for each tree are recorded throughout the season. Sap volume is calculated by measuring, the sap depth in the bucket to the nearest 1/10 centimeter, and using this figure in the formula for a truncated cone to calculate volume. Sap sugar content will be found by collecting sap samples from tubing into microcentrifuge tubes and analyze these within 24 hours in the lab using a temperature compensating digital refractometer. And then the nonlinear mixed effects package in R will be used to analyze this data. And here are some preliminary figures from last season. We finished tapping for this season, but we don't have any figures really outlined yet. And the next steps are to collect the biological characteristics I mentioned earlier this summer, and then we're going to measure tap hole heights and the azimuths to see if there's any relationship between those factors and sap yield, and then of course to analyze the data using R. The second objective, what is the most energy efficient and cost effective method of evaporation? I've been working with 4 producers in Kentucky that are representative of a range of production sizes, and they use varied technologies in their evaporation. Each producer will be sampled 3 times for a total of 12 events. At each one of these events, the wood is sampled, the wood fuel used for evaporation is sampled for weight to the nearest 1/10 of a gram and moisture content to the nearest 1/10 of a percent. 80 logs are sampled at each evaporation event using a convenient sampling. Along with the wood characteristics, syrup volume, sap sugar content, and sap volume were recorded along with relevant time records. The analysis will use the Glimmer package in R. Syrup produced will be the response variable, and the explanatory variables will be sap input, median or mean wood weight, moisture content, and also sap sugar content. The random effects portion of the Glimmer will be used to account for the differences in producer technology. Question 3, what parameters will impact the viability of production found from conducting a cost-benefit analysis? Cost-benefit analysis is a welfare-based analysis tool for comparing courses of action. They compare the status quo versus alternatives, and in this instance, the status quo is no maple production. 3 production sizes based on stand density will be analyzed. The stakeholders will be limited to producers, which means that I won't be including any social benefits or costs. It'll be strictly the producer's costs and benefits. And a sensitivity analysis will be conducted to determine which parameters are most impactful, and these parameters being discount rate, time horizon, and syrup revenues. along with some potentially other ones. The benefits for the cost benefit analysis will be the revenues from syrup production, and the cost will be the required inputs of production, both variable and fixed. Net present value will be the decision rule. The formula for net present value is there on the right. If the net present value is positive, then the investment in maple production should be undertaken. However, if the NPV is negative, then the investment should not be taken. The fourth objective, what are the potential impacts of a maple syrup industry on the state economy? IMPLAN, which is impact analysis for planning software, will be used to run an input-output analysis. A farm budget will be made as an example operation and used to estimate the coefficients used in the IMPLAN software. This analysis will give an output of the effects of maple syrup on the state economy, such as personal income, employee compensation figures, and also multiplier effects. The prospective timeline, the first 2 seasons of tapping have been completed. I conducted some of the energy experiments this spring. However, due to some unforeseen events, including the ice storms, that significantly hampered my ability to visit all of the producers. So that will also continue next spring. This summer, I'll be analyzing the data from objective 1 and 2, conducting the cost benefit analysis and IMPLAN analysis, and then through the fall, working on my thesis with the energy experiments beginning again in the spring of 2022, where I will also continue writing the thesis and thesis defense. The significance of the research is an end goal outlining practical guidance for potential producers. This is composed of recommendations for stand management, recommendations for the most efficient method of evaporation, a timeline for when investment will be recouped and profits made if maple syrup production is undertaken, and then some understanding of the statewide impacts of the maple syrup industry. These are my acknowledgements. Dr. Achudo is my advisor. Dr. Laka is my co-advisor and committee member, and then Dr. Mueller is my third committee member. And Zach Hackworth, he did the first season of tapping entirely by himself, so he's instrumental in this, and he's also been with me each time we go out this season. And Allison Davis, who has helped with the looking at what kind of analyses should be used in R. Here's my literature cited. And any questions?